So thanks very much, guys, to be here. So as Laszlo mentioned, we started to work on network biology, which is now referred to as network biology, about 18 years ago. And of course, this is really sort of totally intermeshed with systems biology. And so there has been a great progress in the last 15, 20 years, right? So I don't need to point that out. But I think that what I would like to tell you is that in the last 2 years, we have to started to work on sort of newer approaches and try to rethink what network biology is really about. So we actually had two things that we got into, which I get to a point, but I just want to give you a little bit of a motivation. So when you think about molecular biology or cell biology, right, it's always cell. Cell. The emphasis is always on cell, and less on the biology. And actually, in reality, I myself came to realize this somewhat relatedly, is that the cell is obviously important, critical, but in real biological systems don't work that way. The cells really don't exist in isolation. There are very, very few cases where a cell exists in isolation. Almost all cases, cells exist as part of a cell ecosystem, a cellular ecosystem. Now, those can be extremely loosely defined. Like, this is not. Do you have a pointer that works in?
Use your mouse.
Oh, the mouse. Okay. Right. That's a challenge because my mouse usually. But anyway, so on the left side, you see actually a bacterial biofilm, right? So these are very transient structures that come together. Different types of bacteria gets integrated with each other. They start to do covering, let's say, surfaces the same day. Immune system, same story. Very, very dynamically changing environment. Now, of course, if you move to a human tissue, for example, like a kidney, there the rules are very strict. Very strict. You have very, very well-defined developmental processes that lead to very well-defined structure. And the reason for that is because the optimal function of the organ can only work in this particular way. So what I'm trying to say is that all this, from the most dynamic to the most defined, you always have interactions between cells. And of course, you also have not only interactions with cells, but also with the environment. So we were trying to look into this, that is there a particular way that this type of thinking might help us to advance network biology? And so I was fortunate actually to set up, to get a grant first of all, a small grant from the NSF for this, and also to hook up actually with a physicist colleague with the name of Hannah Salman, who actually had already established all the different toolkits that we needed for this experiment, or furthering this idea. So now, bacterial taxis, especially bacterial chemotaxis, is an age-old problem. This has like a 40-50-year literature behind it, and this is actually where systems biology to a large extent has started. Like the early work of Leibers that Laszlo and I looked at keenly 20 years ago. These were very important early steps. And I assume that you don't know much about chemotaxis. I don't know much about chemotaxis either, but to very quickly summarize the basic issue here is that bacteria usually move randomly in an environment if there is no particular signal around. And that's shown on the left-hand side here, in the upper left corner. So essentially they have two types of possibilities. They either go straight, swim straight, or they change direction. That changing of direction is called tumbling. Now, you can actually, so there's a relationship, there's a ratio of tumbling versus swimming, and this actually defines what the actual outcome of the movement of the bacteria is. Now, if you have a particular environmental signal present, then there is a bias in this random walk that is built in. And the bacteria starts to move into one direction. It can, let's say, if there's an attractant, it moves toward the attractant. If there's a repellent, it moves away from it. If there is high temperature, low temperature, pH, anything that you want in an environment, the bacteria will react on. Right? So this has been worked out very well, so that the so-called bacterial chemotaxis network is sort of a poster boy for systems biology. But so we need to drill down before I can actually get into the main picture. I need to drill down a little bit. So let's talk about swimming first. So the way that this is really done is that you have essentially a receptor unit, right, on the cell surface that sees the signals in the environment, integrates those signals, and then actually develops an output and drives the flagella, which is actually a motor. Right? So this actually can go either counterclockwise or clockwise rotation, and it's really this. It's really the status of this communicating molecule, the so-called KY molecule, that determines the frequency of counterclockwise versus clockwise rotation. So if you have clockwise rotation, that's induced by the phosphorylated form of this KY molecule, then all the flagella will do their own thing, because in this clockwise setup they don't bundle. However, in the counterclockwise rotation, they bundle. They essentially form a screw, and now they can very efficiently propel the bacteria forward. Okay, so this is all probably what you really know to know about chemotaxis. And then really the ratio of these two things, that the one that defines what's. Okay, so now this is the biology about chemotaxis. So now we want to study this, and we want to build up an environment, and this is what we have spent some time to build up. So this is now we are using microfluidic devices. These are very tiny devices that you don't really see with the naked eye. But the whole idea here is that these devices will allow you to very precisely control the spatial and chemical properties of an environment. And we actually decided to build up this device with multiple purposes in mind, but I'm only really going to talk about one of these, maybe if I have time to. So the whole thing is that we wanted to create an environment where we put different types of traps in front of the bacteria. So they not only are able to exist there, but they also have to go around the object. So we build these things that you can build. These objects are really small. They are various size, 20, 30, 50 micron diameter. So they are small objects. And you can either look at either wild type E. coli cell, or you can use E. coli mutant, or you can build synthetic microbiomes where you put 2-3 different microbes together and see how are they going to behave in this environment. Okay, so the experiment that I'd like to, or the study that I'd like to talk to you about is really this, is nothing less than just single E. coli cells. But we are actually going to study their behavior in what's called a reverse attractant gradient. What it means is that, and perhaps I should go on to this slide, is that we are actually going to, so here is a main channel here. This is where things are coming in, okay? And there's this tiny little inlet, it's only 5 micron wide. And this is where the cells will have to come in, and we have a nuke area. Now the nuke area,
so those squares are not valleys, but they are obstacles, right?
They are obstacles.
I see, so they're like going in between, okay.
Right, so this whole thing at the 3-dimensional, it's 10 micron tall. Mm-hmm. So now the E. coli cell is about 1 to 3 microns. So it's still a sensor. So anyway, so what we can do then is that we can actually have built up a chemical environment inside this way in such a way that you can actually establish a gradient. And I don't want to go through this slide because it's really a supporting slide, but it just really tells you that we can actually measure this gradient and we can characterize the device quite precisely. And okay, so this is the setup, the initial setup. Now, I don't know whether the movie will work or not. I just wanted to show you one movie. Okay, QuickTime is not available. Alright, so that's probably my computer. Well, anyway, so just imagine that we have loaded these cells. The cells are labeled with the fluorescent protein. And now, so we can actually visualize them. And we are now tracing them with an automated microscope.
So each little thing is a cell?
Each little thing is a bacterial
cell. And you can see moving them around.
Right, and we can see moving them around. Mm-hmm. And it would be nice to show it because it's kind of cool, but it doesn't work, so let's go to the next slide. So, so this is these are individual frames from the movie, and you can see at 0 minute on the very top you can still see that we are still loading cells, but by 2 minutes we have changed into a new media which no longer contains bacteria. So we have now a fixed number of bacteria in the microchamber, and now we can add a chemical gradient that is in reverse. So now we are forcing the cells out of the chamber. And what we're actually trying to do here is to measure how quickly the cells are getting out of the chamber. And so you can see that the entry point is on the upper left corner. You can see that. And you can see that the cells are moving up pretty quickly, right? So 2 minutes, 4 minutes, 6 minutes, 8 minutes, like 10 minutes, many of the cells have already moved up. In fact, if you're looking at the curve itself, you can see that this movement, this is time here in minutes, and this is the movement, and you can see it's very quick. It happens very quickly. Now, this is just a control showing you what happens if the cells are not chemotaxis competent. Actually, E. coli cells need an amino acid called methionine to be able to do chemotaxis. If you don't have that in the media, they don't do chemotaxis. So this is what the baseline is showing them.
So they're just randomly moving.
They're just randomly moving that, right? It's not a chemotactic response.
Okay.
So then you can start to do a more detailed characterization. And I would say that the only important thing, or maybe I should actually go back here to, so please read the title here. So it's important because I forgot to talk about that. These receptors come in various formats. So E. coli cells have 5 different receptor types, and these receptors tend to be relatively specific to certain signals. So, for example, the TSR, which is the most common, the most abundant receptor on the cell surface, that is able to specifically sense serine and glycine. These are 2 amino acids, and they can sense it, but for example, the TAR receptor, which is the second most common receptor, is unable to do that. However, aspartate and glutamate is able to be sensed by TAR, but not by TSR. Right? So now we have a toolkit, essentially, that we can start to do, well, what happens if we feed the cells with aspartate, and there's TSR present, or TAR present, or they are absent, or both are absent. That's what it's. So essentially, both genetic as well as chemical inhibition on the pathway. So this is sort of the main slide, I would say, this one, which is actually shows you how what is the migration rate of the E. coli cells out of the chamber. And you can see here is that some cells are not migrating out very quickly at all, other cells are migrating out quickly. And I just would like to draw your attention, for example, for the delta TSR. Right? This is really interesting. The wild type cells are moving out, and with various concentrations of aspartate or glutamate. But you can see that the delta TSR cells are not moving out very quickly in the presence of aspartate. So this is actually a big surprise, because the signal is not through TSR. So if the signal is not through TSR, why are they not moving out? You would expect them to move out pretty quickly. Now, unfortunately, I couldn't find a slide that reverses this, that when TAR is deleted, cells are moving out. So this is an important finding. Now, the other thing is that we can do other tricks. For example, you can also saturate the TSR receptor before you actually putting the cells into the system, because serine, if you have high enough concentration of serine, it's known to block TSR. So on the one hand, you can delete TSR, the receptor, or you can chemically inhibit the receptor. And when you're actually chemically inhibiting the receptor, the effect is even stronger. You can see that the cells are even less prone to move out of the system. Now, this is always a catch of a genetic experiment, because even E. coli cells are trying to compensate. So if you're deleting something from an E. coli cell, you cannot be guaranteed that there is no homeostatic mechanism that tries to compensate for that. So this actually totally in line is that chemical inhibition works better than the genetic inhibition. But it clearly, what the message is the most important one here is that for an aspartate-specific signal, TSR is somehow involved. Now, the second important thing that I will show you later on is that there is also some concentration dependence with weaker attractants. For example, you can take here glutamine. You can see that 250 and 5 millimolar concentration actually changes the way that the cells are migrating out. So this would be the 5 millimolar, and this would be the 250 micromolar concentration. You can see that with the glutamine is known to be a weaker attractant, so they actually exiting at a lower rate. Okay, so this really told us that something interesting is going on because we have a non-TSR specific signal that yet requires TSR. So what's going on in here? And so what we have done is then try to do some additional experiments. First of all, we looked at the importance of cell concentration. So we actually, by this time, we developed a hypothesis. And the hypothesis was that the cells are doing something else. So they are not only going to respond to the chemotactic stimuli, but they are also going to actually create their own stimuli. This was the idea. And so we were trying to generate then some data that would support that. So first of all, we looked at the cell concentration. And so you can see that we have, depending on how many cells we are putting into the chamber to start with, you can see that the number of cells are going to, or the migration of the cells out of the chamber will start to vary. You can see that when we have a low number of cells, they are migrating out at a slower rate than we have a higher concentration of cells. Now, secondly, we ask the question is that is the chemotactic stimulus is really important for the cells to migrate out? And so the way that we looked at that is that we looked in a particular area of the chamber where we know that the chemoattractant stimulus is not yet present. And that's actually measured here, so we are just focusing on this gray area, which is the farthest away from the entry point. Remember that the strongest chemoattractant gradient is here, and the weakest one is in here. And in fact, we are actually using, we can't quite measure aspartate, but we are using a dye that has the exact same diffusivity, which is a fluorescent dye, and that we can track. And so you can see that that's actually, this is the tracking of the dye. And you can see that at the first maybe 8 to 10 minutes, there is very little change. And this particular, we are focusing on the red dot here. I forgot to mention the red dot here. So you can see that even in the absence of the stimuli, that you put in, the cells are already migrating, right? And this is actually, so this is the 200, and this is 20 micromolar aspartate. So what's going on here? We have, how come that you don't see the signal and you are already reacting to it? So that really, these two data together, then we started to think about, well, maybe there is actually, the cells are secreting something in response to the original external stimulus. So we also tried to, so we had some other measurements, and for example, one of the tricks that we tried is to actually increase the number of cells in the chamber. So now we are actually going above 5,000, which is not usually work about 2,000 cells. And what we have found is that, interestingly, when you're applying the external stimulus, the cells are actually not moving that direction. But you can see that at the very beginning, they are starting to coalesce or aggregate in the opposite direction to what we expected. And then after some time, they start to react, and then they are suddenly migrating out of the environment. We also try to put various things. For example, this is trying to find out what may be, assuming that the cells are really producing something, what could be the stimulus. So we were thinking about glycine. Glycine is a TSR-specific signal. It has been described in the literature as potentially being secreted by cells, but when we add glycine to the system, we don't really see a response. Now we also thought about autoinducers. Autoinducers is a group of molecules that varies from bacteria to bacteria, but they are known to be chemoattractant in E. coli, a specific form of it, which is called AI2. Don't want to get into the AI2 biology. That's a big topic in itself. But we actually utilized 2 different AI2-related mutants and found out that they are behaving exactly like the wild type. So we don't really think that AI2 is the molecule. So then we also did then finally HPLC analysis on the XSL medium. So this is a very crude way to determine what the signal might be what the cells are looking. But we either had aspartate gradient on the top or no gradient, as you can see in the bottom. And what we find is that we actually, so aspartate is not surprising to see because that's the input amino acid, that's the chemoattractant. But we are also seeing the appearance of serine within 20 minutes in the culture, which was actually really exciting because that now suggests, what it doesn't prove, but suggests that what the cells are actually secreting is serine, which is the strongest chemoattractant that you can produce. So, so this was in the initial set of experiments. Okay. So because the reviewers asked for it, we also did a numeric. I didn't do it, and I wouldn't be able to explain you the modeling itself. But we also did some numerical simulations with various assumptions. So first of all, that we were thinking that all the agents, bacteria, would be homogeneously distributed at the start. And then we would apply the signal, the attractant, to the upper right corner. So that's your reference point. And we are looking at high density of cells, low density of cells, and two in the bottom, two high density of cells. Now, so this was done by Hannah and Brian Clay from Florida State University. So I'm just reporting what they found. And what they asked was actually a really interesting question because they also asked the question, not only what happens when you signal or don't signal, but what happens when you're actually doing a so-called positional signaling. Positional signaling means that you're only releasing the signal molecule when you are swimming in the right direction. So when you are swimming in the chemoattractant. And interestingly, what this particular simulation shows, you can see it here by 4 minutes. There is lots of aggregation of cells at the exit point, but also shown here on the curve is that if you are if you're assuming that you're only releasing the signal when you're moving to the right direction, then you are actually providing the largest boost for the community as a whole. Now, of course, I would say that there's a caveat here because experimentally we cannot touch this at this point. This is an example where a model is pretty darn useful to drive your thinking, but we don't actually have the experimental toolkits to actually interrogate this question. So this was really exciting. So our working hypothesis right now is that we think, and I will get to that in a second, what the working hypothesis is. Now we are also.
Can I have a question?
Yes.
So you set it up at the beginning that there's actually a network-based explanation, and I guess I keep waiting for that to happen.
Right.
So is this going to come, right?
It will.
Okay, perfect. Okay, all right.
I hope it's good. But anyway, but this is really a biology talk, by the way. So
okay,
it just tries to give a sort of like a. So anyway, now the other things that I already alluded at the beginning is that we have various kind of designs in these microchambers. So what we can also do is to ask the questions that how do the physical obstacles themselves affect the movement of E. coli cells? First of all, you could ask, is there going to be any effect at all or not? And so this is an example where we are actually utilizing a somewhat different, one particular type of design in which the total coverage that the obstacles cover is only 25%, but the size of the obstacles and their density varies. And so we go from 20 to 36 to 15 square micron objects. And we are using two strong chemoattractants here. One of them is aspartate and one of them is N9CG, which is actually a mixture of various types of amino acids. And we clearly see nothing.
What's the size of an E. coli?
It's about 1 times 3 micron.
I see. So these are comparable to the smaller guys, to the smaller.
Yes, right. So I mean, these are 20 microns, so they are much larger than.
I see. So they still see a string versus they don't see this. That's perfect.
So that, but the majority of the work is actually was done in this setup where the size of the objects are exactly the same, 50 square micron, but their density is different, going from 0 on the upper left to 64% coverage on the lower. Now, so this was surprising to us because what we actually found that it depends on how the E. coli cells is actually dependent both the chemoattractant as well as the object. So if you put in the two strong chemoattractants, N9-CG or aspartate, you can see that the cells are essentially behaving very similarly. So they are as if the objects are not present at all. Interestingly, if you're using a weak attractant, such as glutamate, at a relatively low concentration, then the obstacles actually help. They actually help the cells to exit from the chamber. And if you're using the delta TSR mutant, suddenly everything reverts to the same very slow migration out. Now, what happens if you're actually changing this? And we just did a couple of experiments with this, so this is actually not finished. This study is that. So what happens if you shift from 250 to 1,000 micromolar glutamate? And as you can see here, is that now they are getting much closer to each other. So as you are increasing the attractant concentration, suddenly the physical objects materialize. So we are on this second project we have to do much more, obviously. But one of the things that we're actually doing is single cell tracking, which is a very tedious process because we really want to understand the exact movement of the cells. But this is sort of the model that comes out. So we call it active propagation of network activity. That's the key phrase here. And essentially, what we are saying is that the existing chemotaxis model is wrong, and it's probably wrong not only in bacteria but in every organism like macrophages, anything that performs chemotaxis. And so, what is well known is the top portion. This has been known for a long time. This is what Leider and others have modeled, which is that how you are biasing your own random walk pattern and how you build various types of memory elements into that system, like adaptation and so forth. But what we are saying is that there is also a second communication channel, and this is where we are actually releasing a signaling pool from the cell. And that we are thinking that this signaling pool, either in an autocrine or a paracrine fashion, will activate other cells for their own chemotaxis network activity. And we think that this is actually a totally general phenomenon in biology. I mean, this is going to be our working hypothesis. So I don't know if it comes from, but I think this is actually quite important because this really tells you that the cells are really a sort of like social. I mean, this E. coli story really shows that the cells are really social creatures. So they're not only taking care of themselves, but they are also taking care of other bacteria as well. Now, why would you say that? Why did this happen? Well, it's very well known that chemotaxis is an important process for biofilm formation. And if you are chemotaxis, if you cannot perform chemotaxis, your biofilms are not going to behave very well. That's a very active area of investigation in bacteriology, molecular bacteriology. It's well known that their antibiotic resistance, their whole community survival is much weaker if you don't have this propensity for chemotaxis. So I think that we actually have identified a mechanism how to actually bring this biofilm things together, and of course we need to do much more work on this for the future.
So the yellow part is the blue part, right?
So the new part is the blue part. And I would say that
but shouldn't you be getting the signaling molecule also comes in? I see, okay.
Mm-hmm. So now I would say that we know nothing about this part. Okay, so it's a complete a black box at this point. It is also possible that the flagellum motor itself feeds back here. That's possible as well. They don't show that, but it is a possibility. And so it will be interesting to certainly work out details. But it is not really unprecedented, this type of solution in other systems as well. If you, for example, think about the nervous system, right? I mean, the nervous system is set up exactly the same way, where you have neurotransmitters. They are neurotransmitters. If you which incidentally are actually the same amino acids that the bacteria are using. And so we also know in bacteria there's actually like a grand total of 3 papers about this, that the bacteria also has these little granules that contains preformed amino acids. We actually looked at that, and we know that metabolism is not required for this process to happen. So ultimately, I think my time is over because I'm almost talking only for 40 minutes. So I'm going to stop here. I want to tell you that we had a second story in metabolism, which is tumor cell metabolism, that shows the same kind of picture in a different way. There we also show both activation and deactivation as well. And that paper is under revision right now. So I'll stop here.
Any questions? Well, let me actually start myself. So what would it actually take for you to map that out, right? Because the real value of the library papers and the Uriel papers, at least like that level with everybody, was that they did have a precise chemical network what would happen, right? So I guess we need a blue ones, right? So what would it take for you to get that?
It's a good question. I haven't really thought about it a lot at this point. This moves back completely to molecular biology.
Yes, of course.
That you have to identify who the players are.
Yes.
So what I need to do is to find a molecular biologist who is interested in working with us on this issue. But at this point, we are more interested in the phenomenology, because we see that that's actually important to characterize. Okay. So what we are really trying to do is to actually, as I said, the single cell movies are important for us, is to characterize what exactly the phenotypic movement of bacteria is, right? And see how they are behaving in these different environments. Okay. And secondly, because we have the option of labeling bacteria with different fluorescence markers, different proteins, so we can also try to start to create synthetic microbiomes where we are, let's say, put together a delta TSR or a delta TAR or a wild type cell with different and individually follow their movement. Yeah. And the reason for that is because it's very interesting to dissect, what is the role of those cells that are only emit a signal. What are the roles of those cells that are only receive signals, and how are their behavior changing? Are there secondary channels of communication?
Well, you could also do random mutagenesis and see who actually ends up there.
Yeah, that's another possibility.
And sequence them and see who did that, right?
And that's actually very easy to do with the KO collection because we have that. Right? Yeah. So we really have all the cells that are required to do. But that's a really large project. I mean, that's a really large project. So right now we are just trying to really characterize more what's happening in here. And I think that trying to identify this is that if there are secondary communications. The reason I'm bringing this up because some of our HPLC studies are also hint that there are additional communication molecules as well. I mean, the one that we are studied in here seems to show that serine is the predominant one, but we are not convinced that in other conditions that's also the case. In fact, we think that it's going to be we think that the nervous system ultimately developed from this. That essentially the same exact principles operate in a bacteria as in the nervous system. You have essentially pools of various types of neurotransmitters. Some of them are simple amino acids, right? That like bacteria use and some of the neurons are using as well. But it's usually a combinatory release, right? That is finally driving these events. So it's going to be, I think it's really going to be interesting to see how these combinatory releases, if they exist, what sort of phenotype they show on such a simple but basic mechanism as chemotaxis.
The signaling molecule is also a basal nutrient for the cell, right?
Exactly, yes.
So how the cell, is there a threshold that the cell has to achieve? Or is the receptor that plays the role in the choice of using that for the basal nutrient?
So obviously serine, right? So serine is obviously it's the most preferred amino acid actually as a food source. But obviously that's a different thing because breaking down serine in the cell by the bacterial metabolic network is a different function from chemotaxis, which is really using that serine molecule just as a signal of food abundance about the environment. So it's essentially when you have one ligand that plays multiple functions. Right? So on the one hand, it is important for driving chemotaxis. Secondly, it's also a food source. And that's why you are moving toward that in the first place, because you want to clean the serine. But it's the strongest one, by the way. That's known. Serine is the strongest amino acid and the preferred metabolic substrate in terms of amino acids. Yes.
It's a very interesting thought. Phenomenologically, it feels similar to flocking behavior or the swarming behavior of insects and birds and even whales. I wonder, is there a way to perturb the cooperation so that you can show experimentally that, in one condition they do cooperate, and in another condition it becomes chaotic?
Yes. And we are working in that direction too. So that's so right now everything is in a reverse gradient, but when you're looking at forward gradient, things change. And for example, this is exactly what you just described. Unfortunately, I don't have those movies here, or I can't play with them. But we are also looking at that issue. So we are seeing an amazing dynamics in terms of aggregation, disaggregation, waves, various types of patterns appear.
And what's the trigger? What's the perturbing trigger?
Well, the question is that we think that we think that in that situation autoinducers have a role. That has actually been published, that autoinducers are chemoattractant that leads to aggregations. But we didn't get that far with that because that's sort of, we were focusing on this project. We have the initial observations on that.
You will be speaking one day in Pittsburgh, right?
Yes. Yeah, I'll be at UPMC.
Any other questions? If not, let's thank Zoltan.
Thank you.